Hi, I'm Divya Pathak. I'm a principal research scientist at Altasciences. And today I'm going to talk about the poster implementation of a semi-automated bead extraction procedure during analyte purification for high throughput immunogenicity assays. To begin with, biotherapeutics can trigger immune responses, leading to the production of anti-drug antibodies, which may affect the drug's pharmacokinetics, safety, and efficacy. We developed a semi-automated bead extraction and acid dissociation procedure combined with a protein AGL capture. The traditional immunoaffinity techniques using magnetic beads are manual, labor-intensive, and time-consuming, leading to variability, longer assay times, and ergonomic risks. Our goal was to optimize the Bead AGL assay for increased throughput and reliability by automating key steps using the thermoscientific Kingfisher Flex platform. Moving on to the methods, the Kingfisher magnetic particle processor was employed to automate the bead washing and the acid dissociation steps of the assay. The different steps of the method and the formats are presented in figure 2 and figure 3. To go over the different assay steps, on day 1, the samples were acidified and were incubated with a biotin labeled drug for an overnight incubation. The following day, the biotin drug ADA complexes were captured on a streptavidin magnetic beads. And then following an incubation period, the streptavidin beads were washed using the Kingfisher Plex platform, and the ADAs were extracted by acid dissociation, again using the Kingfisher Flex platform. Following acid dissociation, the samples were neutralized and were captured on a protein AGL coated MSD plate. Finally, the detection of the ADAs was performed using sulfotag labeled drug. The key bead parameters presented in figure 3, namely the mixing speed, the soaking time, the mixing time, and the collection time were optimized to minimize bead loss and improve reproducibility. A comparative analysis was performed between manual execution and using the Kingfisher Flex platform for the immunoaffinity purification steps. This analysis involved assessing cut points and precision metrics to demonstrate performance comparability between the two approaches. To go over the results between the comparative analysis, firstly, the control and the individual human serum lot testing demonstrated comparable control responses in the screening and the confirmatory assays. And this was seen with a percent difference within 15% for the controls between the Kingfisher and the manual processing. The variability within the individual human serum lots evaluated on the Kingfisher platform was reduced compared to manual extraction. Yet these results are demonstrated in figure 5 and figure 4. To elaborate, the standard deviation across signals was within 9% for the Kingfisher platform compared to 18% for the manual extraction. And this is for the screening tier. Similarly, for the confirmatory tier, a 9% inhibition on the Kingfisher platform was observed as opposed to the 11% with the manual extraction. Moving on to the cut points. The screening and titration cut points are presented in figure 6. And to go over, the cut points which were obtained were within 25% difference of each other. The confirmatory cut points significantly differed across the two platforms with a percent difference of around 46%. The lower cut points obtained with the Kingfisher Plex platform resulted in improved assay sensitivity, specifically again in the confirmatory tier. The specificity plots presented in figure 7 demonstrate reduced non-specific binding in the Kingfisher Plex platform as compared to the manual processing. This could be attributed to a more effective and uniform bead washing on the Kingfisher Plex platform. Similarly, the false positive error rates were much lower in the Kingfisher Flex platform as compared to the manual processing of beads, which are presented in figure 8. These lower false positive error rates also resulted in reduced number of samples being analyzed in the next year, thus increasing throughput and turnaround time for long-term clinical studies. Finally, moving on to the precision, comparable assay responses were obtained across both the screening and confirmatory tiers across both platforms, and both approaches demonstrated inter and intra-assay precisions below 20%. And these are presented in Table 1. Finally, to conclude, the semi-automated bead AGL assay offers substantial improvements over the traditional manual methods of bead processing. By using the Kingfisher platform, we were able to achieve faster and more consistent bead washing by reducing assay times almost by 30%, increasing throughput by 25%, and enhancing precision. Additionally, it also improved operator ergonomics by reducing the need for repetitive pipetting, making it an ideal solution for large-scale immunogenicity testing. Thank you.